plasma proteins, erythropoiesis, RBCs, WBCs, platelets we have done, blood coagulation we have done. One topic which is mentioned in the syllabus just after erythropoiesis, but that is hemoglobin. We will do that today. It will require 2 sessions, and after that blood groups, another 1 or 2 topics are left, which will take a few sessions, 1 or 2 topics, but it will not, that does not mean that one topic will be completed within 1 session. So, hemoglobin, whatever is written in this first slide, all of you are acquainted. Hemoglobin, obviously it's a protein in red blood cells. So, talking of hemoglobin, this is heme plus globin, you know that also. Heme is a separate entity, globin is a separate entity, and that so together forms the hemoglobin. So, globin is protein subunits. So, what is the main role of hemoglobin in the body? Main role means there are different roles, but the primary role we can say, if oxygen, it does not carry oxygen, oxygen is very little carried in the plasma. It is not possible for oxygen to be carried in a large amounts in the, that means dissolved in the plasma. So, hemoglobin is absolutely necessary to supply oxygen to the, at the tissue level. That means otherwise the person will suffer from something called, or the individual will suffer from something called hypoxia. What is hypoxia will be discussed in detail during the sessions on respiratory physiology, and that is a very important topic in respiratory physiology. For now, hemoglobin carries oxygen, that is the main role, and also a role in carrying carbon dioxide, although proportionately somewhat carbon dioxide more than oxygen can be dissolved in the plasma as well, but without hemoglobin again carbon dioxide carriage will also be hampered. So, just the introductory slides for this. Structure of hemoglobin is heme plus globin. Heme, as you know, contains iron in ferrous or ferric, ferrous form, that is Fe2 plus. Converted to ferric form, it is, if the iron is somehow converted to ferric form, that compound is called I think you have heard these terms. That is a structural variant of hemoglobin, but iron is ferrous form. In this hemoglobin, iron in ferrous form is necessary for all it does. That means the actual role of the hemoglobin to carry oxygen. For that, iron in ferrous form is required, and globin is made up of polypeptide chains. In case of the adults, the adult hemoglobin, most of the adult hemoglobin, which is designated as HbA, has 2 alpha chains, 2 beta chains in the globin part, that is in the globin part. Functions of hemoglobin, this is just in short in this slide, this is just about a summary of what we will discuss in hemoglobin. Some diseases will be discussed in response to, in relation to the variants of hemoglobin. Just again, if we go by the CBME 2024 bio syllabus, it is mentioned as variants of hemoglobin, and in relation a few just 1 or 2 lines about the diseases. Types, what they are saying over here, this is the types HbA, HbA2, HbS. These are some, again some variants, some either some abnormalities like, or HbF is fetal hemoglobin, and the difference is in the globin chains. Fetal hemoglobin has higher oxygen affinity, and functions are mentioned. Normal hemoglobin levels are mentioned, which you already know. These are varying from author to author. Say you can say for adult males is 14 to 16 gram per dl. For adult females it is 12 to 15 gram per dl. So, it's, if it is gram per dl 12 to 16, then just calculate out the total hemoglobin which is carried in a single in the human blood? Just simple mathematics, 12 to 16 gram, say 12 to 16 gram per dl. If I take this value, okay, take a single value, say 15. So what is the total amount of hemoglobin in the blood? This is very basic, very preliminary. How much blood in the body? Say 5 liters. So if 1 dl it is 15 gram, actually in the Western countries they are expressing it in liters. So just multiply that by 5. Per deciliter, if it's 15, then it's 150 per liter. So that's multiplied by 5, it's roughly 750 to 900. So it may be, if it is 600, it comes to 800, 16. So 750, the if we take it to be 17, then it's 850. So 750 to 900 grams of hemoglobin in the body, about 0.3 gram synthesized to, in the, if we talk of homeostasis, 0.3 gram synthesized every hour and destroyed every hour. Okay, 0.3 gram, that is comes to 300 milligram. Okay, so that is just the introductory part. This is how the globin chains look like, helix. We'll talk about that. So before talking of the structure, this is not a slide on hemoglobin, just to make you acquainted. You should know better than me because you have just studied chemistry and come. Pyrole rings. In organic chemistry, pyrole rings. So heterocyclic rings. Heterocyclic rings containing carbon atoms, one nitrogen atom, hydrogen atoms. Okay. So it comes from the Greek word pyros, is that is the origin of the word. So just remember means just see the pyrole ring, how the pyrole ring looks like. So pyrole ring will be for the 4 such pyrole rings will make up something called porphyrin. You will study about a disease called porphyria in medicine. So just remember the name porphyrin for now. So pyrole ring is a single ring. Porphyrins are this combination of 4 pyrole rings, and in the middle and joined by CH, each ring joined by CH to each other, the name porphyrin. Again, if you just remember although it is not possible to remember for each and every term, that will itself become a burden. But if you just remember, in some cases, if you remember the origin of the word, then it is easy to gather the context or where from it comes from. So just porphyrin comes from purple, the absorption burns in chromatography and all. So just remember, for now we will need hemoglobin structure, pyrole rings, the pyrole structure, 4 such rings, porphyrin rings. In the middle there will be a, what will be in the middle? Iron. So this is a structure of this is called metalloporphyrin. So this has just come later. So it should be first metalloporphyrin. Like if it is magnesium in the middle, these are 2 very classic examples of metalloporphyrins. 2 metals in the middle of the structure. One is, if it is magnesium, it's chlorophyll. We are concerned with heme, which is red in color. So this is also why it is red in color, due to the absorption. It absorbs all lights except the red. So these are principles of chromatography and all in physics and chemistry. We are not much concerned about it. So it absorbs the whatever color it won't absorb, that color will be visible. So chlorophyll green in color, heme red in color. The same type of structure, only the metal is different. So this is the structure of heme, not only heme, and this is with all after this is the basic pyrole ring. This is the structure of heme. and this is the structure of deoxygenated. It will hemoglobin, when you call it globin, it will be associated with globin, which is not shown in this picture. Globins are globin proteins after the associated, but they have just shown the oxygen. When the deoxygenated hemoglobin, an oxygen is being attached through this Ferrous moiety. So after oxygen is attached, how many oxygens can be attached to one heme molecule? 4. That is 4 pyrole rings, each will carry 1, and affinity for one oxygen. Once one oxygen is attached, it increases the affinity for the other oxygen. That means more and more oxygen being attached, the affinity becomes more. Okay. Don't think it as a positive feedback cycle. This is not a positive feedback. This is just increasing. This is called cooperative binding. And again, you may know better than me, because these are all concepts from chemistry. You may remember better than me right now. So cooperative binding. So once one oxygen attaches, the other itself increases the affinity for other oxygens. And so how, when oxygen attaches to hemoglobin, this oxygen affinity curve with hemoglobin, this becomes a sigmoid shaped curve. We will discuss about this when, that means you will learn about this when we study respiratory physiology, how oxygen is attached and oxygen is carried by hemoglobin. We will just mention this in 1 or 2 lines now. In respiratory physiology, carriage of oxygen and carbon dioxide in the blood will be discussed in more detail. So this is what the globin chains look like. Mainly globin chains are different types. 2 types are mainly, if you talk of hemoglobin A, which is the most prevalent in the healthy adult, HbA is almost more than 90%, it is almost more than 95%, also more than 97-98%. So that is alpha beta globin chains. If they are alpha beta, these are helical structures. What other helical structure do you know very commonly? We speak of alpha helix. Basic nucleic acid without which living. DNA is a 2 such helices, alpha helix. So multiple these helical structures, globin is one such this protein. How does there are different types of globin, they have differentiated into the alpha cluster. These are again historical origin. Alpha, why did they name as alpha cluster and beta cluster? These are no more relevant right now, not much relevant right now, just the names are from historical origin. Why did they name so? But some genes, the 2 genes on the one on chromosome, the genes on the short arm of chromosome 11 and chromosome 16 are relevant to this. So the main globin gene clusters are located on these 2 chromosomes. And how this combination alpha beta, so how many are there? Alpha, beta, gamma, delta and epsilon. Okay. So these are the, you have to know this. So alpha, beta, gamma, delta, epsilon and zeta, 6 types in the 4 genes. Alpha, beta, we will come to this. Alpha, beta, gamma, delta, just you have to name this. If it is, the name is written like this, especially in the MCQs, either the sign may be given or the name may be given. So you have to identify. So alpha, beta, gamma, delta, Greek letters, epsilon and zeta, these types. So, and the combination is random, but usually it does not. Combination, which two will be of one type, 2 will be of another type among the 4. The combination, different globin chains, they are directed by the individual genes combined in random fashion. But in the healthy, if it is physiological, then it is seen in the fetal, which predominates the most, the HbF. Then in the adult, it is HbA. HbA2 is another variant. We will talk of this. This is showing this is not very important for you, just see this alpha chain, how it is. Alpha chain, see, during this is measure, the x-axis is gestation in months, and after birth it is the age in months. So before the birth, what is happening? Alpha chain is being produced 40%, very less now it is becoming 50% of alpha chain. So at the adults, after a few months, after 3 months, the alpha chain is all types of hemoglobin till now known, the alpha chain is common in all types of chains. How do you? This is 50%. So, 2 alpha chains at least in all forms of hemoglobin. Before, after, say roughly from 3 months. Before 3 months, these epsilon and gamma chains are more prevalent. Epsilon and zeta chains, then gamma chain, and in the adult, see, in the adult after birth, the beta chain is taking a upper hand and alpha chains are already there. So, it shows in the adults it is alpha 2 beta 2. Do you get this? So, this is not very important for you, I repeat that you do not have to draw this anywhere or something, just, but as we know alpha 2 beta 2, see the production, the rates of production of these chains vary. This is from a very standard textbook. So, you do not require it. So, delta chain after birth, say up to 6 months like this, but the alpha chains and beta chains gradually take the upper hand. So, hemoglobin A is alpha 2 beta 2. So, this shows, this is the percentage of total chains. So, this shows the 3 months onwards alpha chain is 50%. So, alpha is common 3 months onwards. Okay, so those are the heme and globin chains. This is not on hemoglobin, just as I told, don't make a confusion during the exams, especially when you have to think of many things, there are confusions. This is not anything about hemoglobin, just globin and globulin and separate terms. Like I told in the last class, hemostasis don't confuse with homeostasis, completely separate entities, completely separate meanings. So, hemostasis and homeostasis are different, globin and globulin are different. Globin are whatever we are talking of now. Myoglobin is another protein like hemoglobin which can carry 1 oxygen. Okay. It is present in muscles. Myoglobin cannot carry 4 like hemoglobin, it is present in muscles. So, globin is the superfamily of heme containing globular proteins, but globulin is a whatever plasma proteins we have talked about globulins already, and they are separate terms, do not confuse during the exam. That is why I just put this slide over here. This has globulin has nothing to do with this hemoglobin discussion. Like hemostasis is cessation of bleeding, okay, whatever we talked of in the last 2 classes, hemostasis, and homeostasis is maintenance of the internal milieu, the internal environment of the body. So, those are 2 separate terms. We are seeing students are, when they are asked to write about homeostasis, they are describing the hemostatic pathway, and this has happened in the last university exam, just in the last university exam which took place in October, November. So, do not confuse globin and globulin, these are 2 separate terms, and globin is a much smaller protein, it is water soluble, but globulin is not soluble in pure water, and all these are the properties. You just, this is a image from the internet itself, you just type globin versus globulin, you will get this. But remember, I just mentioned it so that you do not confuse. Remember this and do not confuse, and that is the importance of attending classes. In the books, you will just, when you read about this topic, no one will write over there globin versus globulin, but just keep and watch over these things, do not confuse. That is why I put it over here. Hemoglobin synthesis in short, it is from, you will again study in medicine, and I think you are studying biochemistry about the, how the about these pathways. So, succinyl-CoA plus glycine leads to pyrone, and pyrole links to porphyrin, it is protoporphyrin, then iron attaches, and then the heme is formed. So, heme and globin together, it is hemoglobin. Okay, and now the detailed structure will just show it over here, but I think that these different biochemical, these pathways you will also study in biochemistry, the formation of different these structures, compounds. Okay, so succinyl-CoA plus glycine pyrole, this is delta amino levulinic acid, this 5-amino, this is delta amino, also written as DALA. DALA means the D is, instead of D there is a delta sign, then ALA, amino levulinic acid, then very simply, then it forms with the, from the pyrole ring, the protoporphyrin. So, protoporphyrin plus iron, it will be the heme attached with the globin chains, it will form the hemoglobin. And where does the hemoglobin synthesis take place? Along with the, we have talked of erythropoiesis, Yes, obviously. Along with the hemoglobin, it starts appearing. We saw this slide during the session on erythropoiesis. So, it starts appearing intermediate normoblast stage, at the intermediate normoblast stage of erythropoiesis, okay, and it continues and increases in amount as erythropoiesis goes on. Starts at the, so this may be a potential MCQ, it starts at which stage. So, hemoglobin is formed, whatever, if it is, you are asked to mention the sites of hemoglobin, it is formation, it is formed inside the maturing erythrocytes. So, it is the same as the sites of erythropoiesis, yolk sac of then liver, bone marrow in the adults and so on. So, in the embryonic stage, yolk sac and all. So, this is if it is the in the this point it is written, if it is beta chains and alpha chains, then it is hemoglobin A, and if it is something else with the alpha chains, mostly 3 months onwards alpha chains is constant. If it is something else with the alpha chains, then they are different names. So, this is in short, see, heme part is synthesis. This is the synthesis of heme, again succinyl CoA glycine, delta amino levulinic acid, that comes to cytoplasm. After the uroporphyrinogen, corpoporphyrinogen, these structures you will read in biochemistry, also it will be discussed in biochemistry. And this corpoporphyrinogen, again this goes into the mitochondria, the last steps, last final steps are again in the mitochondria of the maturing RBCs. The heme part of hemoglobin, does adult RBCs contain mitochondria? Sorry, adult means mature RBCs? No, it is the maturing RBCs. So, maturing RBCs lose their nucleus and mitochondria just before the formation of the penultimate stage, the reticulocyte. We studied that during erythropoiesis, but during maturing, this mitochondria is important. These steps occur in the mitochondria. So, heme part of hemoglobin together in the mitochondria and the cytosol, globin's part completely in the cytosol, that is the cytoplasm. So, from the pro-erythroblast stage, or they are writing the pro-erythroblast stage, but actually it is the you write it as the intermediate normoblast stage to the full maturation, to the still the reticulocyte becomes the erythrocyte, hemoglobin synthesis goes on and it increases. So, this is again the pathway which we was talking of, with all the enzymes and all, which will I think we will study in biochemistry, the pathway of formation of this porphyrinogen and porphyrin, and starts with glycine plus succinyl CoA, which is the rate limiting step, this one, the first one, the ALA synthase, the formation of the ALA, that is the rate limiting step in this formation of heme. So, the first step. Okay. So, all these ALA dehydratase uroporphyrinogen 1 synthase, I do not think from the examination point of view, I do not think this will be asked in the, as of now, physiology and biochemistry was actually taught as a single subject in the earlier days, not even when I studied MBBS, but before that, much before that. So, now they are separate. If it is in your biochemistry curriculum, then they may ask about the details of the steps. I do not think in physiology now the type of questions they are setting. So, they will not ask you to write about this thing in physiology, but this is a part of heme synthesis obviously. In the physiology question paper, you will mainly get short notes based on these clinical applications of different concepts, short notes, direct short notes. Say if heme synthesis is mentioned as a short note, then if you know about this, write as much as you can. Okay. So, mentioning the enzymes and all, but probably write the steps mentioning the enzymes and all, they will not come as the again, but MCQ may come, say MCQ, they may give 1 or 2 MCQs like which is the rate limiting step and all, but probably write the steps of the heme synthesis, how porphyrin is formed. This will may be a question in the biochemistry paper, but not in the physiology paper. Mainly the National Medical Commission is also, that means they are also influence, that means directing that question should be based on clinical orientation. Okay. So, maybe we are talking of some diseases in every topic. So, in the first year of your medical school, you are probably not required to diagnose anything. So, a disease related to this hemoglobin is thalassemia, or a disease related to a coagulation disorder is hemophilia. So, you are not required from the question, you are not required to diagnose anything. That is a part of your medicine curriculum, and that may be that type of question may be set in medicine. The diagnosis will be given, but then the relevant questions will be asked on the physiology aspects of those topics. Okay. So, you will not be asked to diagnose probably peptic ulcer, although sometimes they are setting it like that. So, some are easy, but still again in my opinion, but in the opinion of most faculties, peptic ulcer, it should be, peptic ulcer is already discussed. So, if you go through your GI syllabus, then you will see they have mentioned some names of some diseases, peptic ulcer, Hirschsprung's disease, maybe pancreatitis also, but you will not be asked for the diagnosis probably, that will be given in the question, but the relevant questions, exocrine functions of pancreas and all, in case of peptic ulcer, how is acid secreted and all. So, that formation of HCl in the stomach, these will be asked, the basic concepts. And if you go for say MD course in medicine also, in any subject there are basic one paper just for the basic concepts. In MD medicine, one paper is only for the basic concepts covering physiology, pharmacology, pathology. So, in the first year of medical school, diagnosis is not that important, diagnosing yourself, but you have to know the concepts. So, the basis of studying this medicine is physiology, pharmacology, pathology. The basis of studying surgery is anatomy and pathology, of course, also pathology for so these are the again important for, I think, historical point of view, and maybe some MCQs mention these. So, Gawar 1, Gawar 2. So, we told before 3 months. So, see, there is a Gawar 2 type, epsilon 2 and zeta 2, 2 chains epsilon 2, zeta 2. And what does the name Gawar, Portland, these names come from? Portland is also mentioned, yes. Portland, this comes from, again, Gawar was maybe is a place, I don't know, it is a place in London, UK. Portland is also in some country, I just can't recall at this moment. So, according to the names of the places where they were discovered, they named it, just of historical importance. But again, if they mention in MCQs, then you have to know. So, Gawar 1, it's alpha 2 epsilon 2, Gawar 2 zeta 2 epsilon 2, but in most Portland is zeta 2 and gamma 2. So, 4 chains, these things you will get in the books, but just have a note, because days of MCQs, if they ask anything. And so, typical fetal hemoglobin times, again come back to this slide. So, rate of change. So, gamma epsilon, they within 3 months physiologically, at least normally, within 3 months gamma and epsilon chains will no more be formed. Okay. So, these must be fetal hemoglobins. So, alpha 2 beta 2 is adult hemoglobin, and another variant of adult hemoglobin which we call as A2 is alpha 2 delta 2. See, delta 2 is produced just before birth, delta chains are starting to produce and then going on, but in small amounts, the amount is small. So, it is, just see from here, it is maybe in the range of 5% or something. So, HbA2 will be very less, even if present, that is physiologically. If in pathological conditions, in disease conditions, these may alter, but physiologically alpha 2 beta 2 is the most prominent in adults, beta 2 alpha 2 delta 2 HbA2. And again alpha 2 gamma 2, which is fetal hemoglobin, to some extent it is present in the adults. You can again see gamma chains, although they decrease after birth, they continue to be formed to some extent. See, in the gram it is not coming to the this 0 line. So, very little gamma chain continues to be produced throughout life. So, although most of it is hemoglobin A, but somewhat is, some is alpha 2 beta 2, some is A, some is alpha 2 delta 2, that is A2, and very little is also alpha 2 gamma 2, that is F, because gamma chains are still continue to be produced maybe throughout life, okay, but very small amounts, as you show from that the rates of synthesis, that graph. Functions of hemoglobin are pretty easy, that in the context that it carries oxygen and carbon dioxide, you already know about that. It imparts red color to the blood. I told you why, because that red light is not absorbed and it shows. So, hemoglobin imparts red color to the blood, the RBCs, that is why hemoglobin are housed in these cells, which are called the, so they are called the red blood cells. That red color is due to the hemoglobin. It carries oxygen and other gases, obviously oxygen and carbon dioxide are the main ones which we talk about, buffers the blood pH. So, hemoglobin has an important buffer, and it is the most important or most abundant single buffer, that is a single molecule is acting as a buffer. Buffer, you know from chemistry, what is a buffer? Some people are saying no, buffer word you haven't heard, you should have heard the word buffer, okay. So, mainly we talk about this. Buffering can be, that means buffering means in if you go by the English term, it is neutralizing, like that, but we mainly talk about this in the context of acids and bases, okay, acid base. So, buffers blood pH. Blood pH is something which is to be maintained in a very, that means narrow limit. So, if you say sometimes, say we talk of we will know about the levels when you in biochemistry, and then you will require it all throughout your life as a practicing medical professional. Say sodium levels, say 135 to 145, whatever is the unit millimole per liter and all. So, these things, if it comes down to 125, say even 110, say 112, that is called hyponatremia, that means sodium is coming down in the blood, but still symptoms will take some time, or in some people it is seen up to say 110, 115, there are no symptoms. Still, if it goes still less below that, then there will be symptoms. But pH is a thing which has to be mentioned in the very narrow range. Even 6.5 is very bad. 6.5 is actually, it may become incompatible with life also. So, it is, so the pH has to be maintained in a very narrow range, and also much alkalinity, that means above 7, above 7.5. So, we do not hear of some pH, a person is living, we have taken out a blood picture, okay, in the laboratory, you do not never seen, you will never see pH of 8. You may think 7, 8, it is a small difference, but actually pH is very sensitive thing. It has to be maintained within very narrow limits. There are other buffers we will talk of in the subsequent physiology classes, but hemoglobin is the most abundant single buffer. Singly, it is acting as a buffer. Source of physiological active catabolites, genetic resistance to malaria, regulation of blood flow is still, we will talk about it. But source of active catabolites, I do not know whether this can be called as a function. Hemoglobin is breaking down, we will talk about breakdown of hemoglobin and form some substances, biliverdin, bilirubin, this globin part will be, again heme part will be separated, globin part will be separated, the iron will again be reused. So, they are writing this slide that source of active catabolites, but this is just, I do not think, do not write it as a function, although they are writing. These 4 can be written, but source of it is natural that heme will break down and form the catabolites. So, it, to me it sounds somewhat foolish that whatever is natural, it has to be, it is not that it is performing that as a function. Those catabolites are required, they have functions in the body, but heme, it is not that heme, it is a hemoglobin is itself controlling it, hemoglobin is breaking down, so those catabolites are formed. So, you may write, but that is not a very important as a function. Again, genetic resistance to malaria, we will talk about it next day. So, abnormal form of hemoglobin like HBS, they have genetic resistance to malaria, but that should not be called a function of it. Do you get it? Whatever is happening due to some disease, they are becoming genetic resistance to malaria. That is an advantage of having something called sickle cell anemia, but that should not be called a function of hemoglobin. Okay, so concentrate mainly on these four. So oxygen delivery to the tissues, again it will be discussed. Oxygen carrying capacity, how is oxygen carried more in respiratory physiology. For now, just know 1 gram of hemoglobin carries 1.34 ml of oxygen roughly, and this in the practical examination just which you will be facing in the internal assessment, maybe dates are being finalized. It will be next month, I think, somewhere sometime in February, in middle of February. So there in the hemoglobin practical, this where we have practical, in the practical portion we estimate hemoglobin. So I have mentioned in the practical classes, questions won't be direct always. Okay. So find out the or calculate the oxygen carrying capacity of blood. Although respiratory physiology is still now not discussed, this simple thing, 1 gram of hemoglobin carries 1.34 ml. You have to estimate hemoglobin in the laboratory and multiply it by 1.34. So that figure is, that is the amount of oxygen carried by 1 deciliter of blood, because you calculate hemoglobin or estimate hemoglobin in gram per deciliter. So multiply by 1.34 by 1 deciliter or 100 milliliter of blood, that amount of oxygen is carried, and in the question it will be mentioned that ignore dissolved oxygen. Okay, because very little, but still there is some, or the dissolved oxygen may be mentioned that this much is dissolved oxygen. So then you have to add that to the whatever you calculate, you have to add that. Calculators will not be provided in the exam. So some figures, if it comes like, say your estimate comes to 13.8, we give credit. That means credit is awarded. If you just write, you have to show the logic, not the final figure is not always required. Calculators, mobiles are not allowed. So you write 13.8 into 1.34. That will suffice. Okay, you don't have to calculate. If it is difficult to calculate in the mind, so you don't have to write the final figure. But if it is 13.6, if 12.6 into 1.34, if it is something a whole figure 14 into 1.34, then it is more or less possible to calculate. But even if not possible, you will get credit if you just show the logic. So it is hemoglobin, blood's capacity, hemoglobin into oxygen capacity of hemoglobin. When oxygenated, again we will talk about 2,3-DPG. That means we will learn about 2,3-DPG when we discuss respiratory physiology. Again, it is an enzyme present in the RBCs. So that is 2,3-DPG pushed out. Beta chains are more, this is involved in the carriage of or attachment of oxygen. When oxygenated, beta chains are moving closer. That is again a physical or chemical property. So 2,3-DPG is one of the factors which is influencing O2 affinity. Again we will, this O2 affinity, how the O2 hemoglobin O2 curve, when oxygen is attached to hemoglobin, how the curve is looking like, sigmoid shaped curve, we will discuss in during respiratory physiology. Okay, and very important topic that is asked in the from the examination point of view also. So most of the oxygen, again only maybe 2% is dissolved in plasma. So 98% which has to be carried. So hemoglobin is absolutely essential for carriage of oxygen and delivery oxygen to cells. Cooperative binding I was talking about. So one oxygen molecule being attached, the affinity for others are increased. 2 is attached, still the affinity still increases. So the pace increases. As soon as 1 is attached, the pace increases. Like last day we talked about something in coagulation. Do you remember something? Just relate things when you study. 9 months, 10 months, 8 months, now maybe 8 months are left. It is very short. The topics, simply the huge number of topics to be covered including the 3 subjects is very short. Do you find much tough things here in today's discussion? Very tough? Sir, I can't understand, anything is not possible. So that is not the problem in MBBS. In the MBBS course, that is not the problem. Problem is the amount, the volume of the syllabus, the volume of the topics. So whenever you study something, try to relate things. Here we are saying cooperative binding. As one oxygen attaches, the affinity, similar thing we discussed in coagulation. Remember factor 12, okay? Just try to remember. If I tell you, then Just try to remember, just go through previous days notes, or just after this class take say 2 minutes. If you have the book, just turn around the book. Factor 12, what happens? In coagulation, assuming we have to complete this discussion, but those compounds I can show it from, okay, leave it, let us complete this discussion. So, how does the factor 12 is the starting of the which pathway, extrinsic or intrinsic? So, factor 12 to factor 12A, this starts at a slow pace, then something happens and then the pace is increased. We talked of this. So, just recall, we will end this discussion first. So, cooperative binding increases. This is about attachment of O2, again CO2 will again discuss in respiratory physiology. As CO2 is attached, a compound is formed which is called carb amino hemoglobin. And just again for MCQ purpose, or if you can write it as a part in some short note, oxygen is attached to the heme part, carbon dioxide attached to the globin part. That means the globin chains are attaching carbon dioxide through this amine group. So, it is called carb amino hemoglobin. You do not require the detailed structures, nobody will ask you to draw these structures, I do not know, probably not in biochemistry also, but just know carb amino hemoglobin, because this is the amine, these amine groups in the globin chains are involved in attaching carbon dioxide, and oxygen is attached to the heme part through the ferrous entity. So, if it becomes ferric entity, then attachment will not be possible, that is called methemoglobin. So, in a way it is abnormal hemoglobin which is not allowing carrying carriage of oxygen. Buffering is through the, again you will get in books, but you may just note it down, through the imidazole groups of histidine residues of hemoglobin again. And within a, again see 7.0 to 7.8. So, I told you we have probably never heard of pH 8. So, it is to be mentioned in a considerably narrow range. So, these imidazole groups are amino acids histidine, they are responsible for this buffering. So, as oxygen in the RBCs, as oxygen dissociates from hemoglobin, this buffering capacity, or as oxygen exceeds from RBCs, H plus is also removed by hemoglobin, that is talking of oxyhemoglobin, but more important or more potent buffer than oxyhemoglobin, that is oxygenated hemoglobin, is deoxygenated hemoglobin. I will mention another thing over here, I just recalled that never confuse between homeostasis and hemostasis, never confuse between globin and globulin. Hemoglobin, when attached to oxygen, it is oxygenation. Very wrongly sometimes this is not oxidation, just it comes to our mind that oxygen is being attached, so it is oxidation. It is not oxidation, it is oxygenation of hemoglobin. Oxidation is whatever we have read in biochemistry, or sorry, chemistry, also in biochemistry of course, but biochemistry, concept of chemistry, whatever, electrons removal, oxidation, reduction, that is oxidation. Hemoglobin attachment to oxygen is oxygenation, and always not that use the terms oxygenated hemoglobin, deoxygenated hemoglobin, oxidized hemoglobin, or reduced. There is a term called reduced hemoglobin, very important term, which is deoxygenated hemoglobin, but the process is not oxidation reduction, it is oxygenation and deoxygenation. Reduced hemoglobin, where did we mention reduced hemoglobin? Whatever we have studied in physiology till now, where did we mention reduced hemoglobin? Think of the clinical classes, just a hint. Just clinical class, clinical physiology classes, at least who have joined earlier during the discussion of Sally's method. Is there any mention of reduced hemoglobin over there? I do not think so in Sally's You should call it not the Sahal's method, but the Sahal's acid hematine method. To be correct, Sahal's acid hematine method, because acid hematine, that compound is formed, that comes within the name of the method. Don't just say Sahal's method. But we mentioned it in the clinical classes. General physical examination, where cyanosis, the definition of cyanosis. We will come to that. Cyanosis is reduced hemoglobin above 5 gram %. Okay, so that is cyanosis, definition of cyanosis, which will be discussed again, but cyanosis we mentioned reduced hemoglobin in that part in the practical classes. So that is reduced hemoglobin, but the process is not oxidation reduction, it is oxygenation and deoxygenation. And how does hemoglobin, we talked of those four, how does hemoglobin regulate the blood flow? See this, I told you, don't just mention, you may mention, but strictly speaking it should not be active catabolites, genetic resistance to malaria, these should not be called as function strictly. So, but regulation of blood flow, it has a role through nitric oxide, it can bind to nitric oxide as well, and nitric oxide, I do not know whether it has been mentioned to you till now, it is a vasodilator, that means it dilates the blood vessels. So, when it releases oxygen, along with that releases nitric oxide also, and it is a physiological role, how it is releasing oxygen to the tissues, hemoglobin, it releases nitric oxide. So, vasodilation occurs, oxygen carriage is becoming easier. Okay. So, that is along with oxygen, nitric oxide is also released, you remember it simply like this. So, this increases blood flow and enhances oxygen delivery. The oxygen is this nitric oxide is attached to oxygen as a compound in HbSNO, just I can't recall the full form of SNO right now, I will tell it to you in the next session. SNO, something, the SNO, HbSNO, I will tell it to you in the next session, what is the full form of SNO. So, it remains like HbSNO and then it is released. As these four oxygens are being released, this nitric oxide is also being released, and nitric oxide induces vasodilatation, so oxygen delivery is easier. So, as nitric oxide is involved in vasodilatation, so this is the regulation of blood flow also. As the vessel dilates, blood flow is regulated. That means in this case, during constriction, blood flow becomes lesser, during dilatation, blood flow becomes smaller. And if oxygenation or deoxygenation of hemoglobin is not proper, then delivery of oxygen is also, firstly, the oxygen, if it is deoxygenation not proper, how can it deliver oxygen? And there that vasoconstriction will also not allow it. So, it is like a double whammy, because oxygen is not being released and together nitric oxide should be released, that is also not being released. So, one of the factors which could dilate the blood vessels is compromised. So, through constricted blood vessels, delivery of oxygen, blood flow, all are delivery of, in that way where delivery of nutrients also get hampered. So, along with carriage of oxygen, carriage of carbon dioxide, buffering action, we should also mention regulation of blood flow as one of the important functions of hemoglobin, but probably not those resistance to diseases and all, those are just pathological conditions. So, or this source of active catabolites, they are probably not functions directly, they are natural processes, hemoglobin is breaking down, so those catabolites will be formed. So, this is about the synthesis, structure and functions. So, we will have to just discuss a few lines about catabolism of hemoglobin or breakdown of hemoglobin and the variants, and in relation to that some diseases, that will be done in the next session.